Hi, everybody. Good afternoon. So, yes, I'm Giulia Babbi from the University of Bologna, the Biocomputing Group. And I want to thank the organizer for giving me the opportunity of presenting my work, that is analyzing multifunctional proteins with multifaceted-ProtDB. As you may know, the problem of multifunctionality has been addressed many times in last decades in the literature, leading to many different definitions of multifunctionality covering different aspects of multifunctionalities. For example, promiscuous enzymes that are enzymes having multiple substrate, or multi-class enzymes with multiple catalytic activities, or pleiotropic proteins when a protein is associated with many different phenotypes, or moonlighting proteins when different functions insist on the same protein domain. Here we propose to collect all these different type of multifunctionalities in proteins under a unique label that is multifaceted proteins. And we believe that this topic, multifunctionality in protein, is a great area of great interest. And is, with this work, we want to try to annotate those proteins, to collect them and annotate them, and finally trying to dissect the complexity of their biological processes and their properties. So first, we collect multifunctional proteins in human. We search the literature and SwissProt. We use many different keywords to try to catch all new reviews and also proteins in SwissProt with a high level of annotation. We filter for protein having a level of annotation of 4 or 5 over 5. The keywords that we use are multifunctional, multitasking, and all the different definitions that we already saw in the previous slide. And finally, we check manually, we curate each entry manually to assess its multifunctionality in the literature. And then we also search public available data sets that are very specialized, especially in moonlighting proteins, MoonDB, MoonProt, and MultitaskProtDB. They collect about 50, 100, and 180 different proteins, and when merged, they collect about 250 proteins with a very small overlap of just 15 proteins. And finally, we collect also enzymes, and though with multiple catalytic activities, both covering the definition of promiscuous and multi-class enzymes, and there are about 680 different enzymes. So at the end, we collected 2,115 different multifaceted human proteins. As far as we know, this is the largest collection of multifaceted human proteins nowadays. So besides the increased number of reported human multifaceted proteins, the contribution of our research stands also in the curated level of annotation of those proteins. We report both molecular features and systemic features. We started reporting the gene and protein name, the protein sequence, the function, the catalytic activity as a AC number if it's present, the protein structure if a PDB is available, and the AlphaFold 3D model, gene ontology terms, and also annotation about protein families and domain as linked from Interpro and PFAM. And we relate these molecular features with systemic features like biological pathways from reactome, tissues of expression and cell type of expression from Human Protein Atlas, and also phenotypes from Human Phenotype Ontology. We curated and integrated the disease annotation of these proteins using different resources. One is OMIM for genetic diseases, but also the ontology from Mondo and ICD-10, and also rare disorders from Orphanet. So at the end, we release our resource that we call MultiFacetedProtDB, a database of human proteins with multiple functions. It has been published in the last NAR database issue. This is the link that links you directly to the web server or the QR code for the publication. So in the web server of MultiFacetedProtDB, there is the possibility to search for a protein using the protein name, the gene name, and many different entry types, looking for a confirmation of its multifunctionality. And if the protein is present, the user will retrieve a result page with all the different annotation, the molecular annotation, the systemic biology features summarized in tables with useful links. There is also the possibility to search with advanced search for a set of protein having similar features, having the same features. For example, we try to search for protein having multiple annotation for diseases in different ontology. And there is also the possibility to connect these different features with logical operators building complex searches. And at the end, the user will retrieve a list of all the proteins with those features, and there is also the possibility to download it. So here we report some general dataset statistics divided into not enzymatic proteins and enzymes, because at the end, 58% of our dataset is made by enzymes. We see that 72% of the proteins have at least one PDB structure, and almost all the proteins have some annotation for protein families and domains from Interpro and PFAM. 83% of the proteins have some annotation in a reactome, and 40% of the proteins are associated with disease as expressed by Mondo identifiers. We then decided to investigate the characteristic of multifaceted proteins from different points of view. So here we show the distribution for tissue of expression of multifaceted proteins expressed as percentage compared to the annotated human proteins in general. So in blue, you may see the multifaceted proteins, and in orange, all proteins as a background. We derived this annotation from the Human Protein Atlas, considering only high and medium level of expression. And we may see here that for this characteristic, there is not an emerging signal as long as the multifaceted proteins are spread all over the tissue in the human body, following the background distribution. For this characteristic, the subcellular location association, we see instead a sort of signal. This is the distribution for subcellular location of multifaceted protein percentage, always in blue, compared with the background in orange. We derive this annotation from Swissprot, and we see that the 50% of our data set consists of cytoplasmatic proteins, and about 40% of the proteins are nucleic proteins, conveying 900 in one case and 700 proteins in the other, respectively. And both these percentages are higher than the background. Here we see the reactome association. We take advantage of the hierarchical structure of reactome, and we decided to see the distribution of multifaceted proteins association with reactome roots, compare always with the background in orange. We may see that there are some roots of biological pathways that are more frequently associated with multifaceted proteins, especially metabolism, having an association with the 35% of our data set, and also immune system, but also metabolism of proteins and signal transduction. And finally, the disease associations. We decided to use the ICD-10 classification. ICD-10, as I said, is an international classification of diseases that is always used by physicians in the hospitals. And we decided to use this classification, this ontology, because there is the possibility to sum up different disorders using the chapters that collect similar diseases or diseases insisting in the same system in the organism. So here we may see that some multifaceted proteins are associated with congenital malformation and chromosomal abnormalities, but also with diseases of the nervous system and endocrine, nutritional, and metabolic diseases. One of the possible applications of our research is to use this data, these curated entries, to link the molecular level of annotation of multifaceted proteins with their system biology features. And this is of particular interest in the case of reactome pathways associated with different diseases. We were able to associate 1,700 different proteins with reactome pathways, and 800 different proteins with Mondo identifiers for diseases. A similar number also for orphanet, a similar number also for ECD10, and also a similar number for phenotypes from human phenotypes ontology. So this is of particular interest in the case of multifaceted proteins because often when these proteins are associated with diseases, they are associated with multiple diseases at the same time. So as you can see, we have 626 different proteins that have each multiple Mondo annotation. And about 60% of the protein that have an orphanet annotation have multiple orphanet annotation for rare diseases, a similar percentage also considering ICD-10 classification. So we can use this association to relate the disease with the biological process that is described in the reactome, and this may help suggesting possible mechanisms of disease insurgence. So as a case study here, we propose apolipoprotein E. Apolipoprotein E is encoded by the gene ApoE. It is a protein associating with lipid particles. This function has been studied in deep, and this protein is related with 11 different reactome pathways. 4 of them are part of the reactome pathways of transport of small molecules, namely chylomicron assembly, chylomicron remodeling, chylomicron clearance, and HDL remodeling. Considering these 4 pathways, it appears clear the link with the disease called hyperlipoproteinemia of type 3, that is a rare condition that presents xanthomatosis and also premature coronary and vascular disease due to accumulation of chylomicron and very low density lipoprotein residues. But this protein, and in particular the variant 4, ApoE4, has been associated with Alzheimer's disease of type 2, that is a specific type of Alzheimer's disease with a late onset. And recently, this protein is being associated with the reactome pathways of amyloid fiber formation. And so this association may help in explicating the mechanisms of disease insurgence. And what is of great interest is that this protein is still associated with 7 more reactome biological pathways and with 2 more diseases. And maybe further studies in this direction will help in relating the biological pathways to the disease mechanisms of insurgence. So concluding, we curated the widest collection of multifaceted human proteins, and this collection is freely available online and this link. And I want to stress a little bit more on the fact that this collection has been curated both in the term of checking the multifunctionality and also in their annotation from a molecular perspective and a system biology perspective. Here we detail several associations among multifaceted proteins and diseases, biological processes, tissues, and subcellular location. We plan to use this data to dissect the complexity of the association among multifaceted proteins and diseases. We think that this may help in understanding disease insurgence and molecular mechanisms. And finally, we also think that this could be a useful data set to study comorbidities, as long as multifaceted proteins are so often associated with multiple disorders. They could be used to study possible comorbidities. And please, if you want to try our research, you may just scan this QR code and you will be linked directly to it. So I want to thank all my co-authors from the Bologna Biocomputing Group, especially my PIs, Pierluigi Martelli and Rita Casadio. And I want to thank you all for the attention. I'm happy to take any questions.
Hello.
Hi.
I have a question on the oligomeric states of these proteins, if you annotate them as well. Because with ApoE, of course, it works as a multimer. So just I was wondering if you annotate also the oligomeric state.
Yes, a good point. No, actually, the oligomeric state is not strictly annotated, I mean, we link the resource from UniProt, so if it is present as a key for the function, it's been written in the function field, so it's being reported, but no, we didn't check the amount of protein with this particularity.
Yeah, so my question is similar. So for multifaceted means it has multiple functions, but how do you actually define a function? So for example, in the page you just show us, I just checked the 2 random proteins, and both of them, they indeed have 2 EC numbers, but the 2 EC numbers are different only by the last digit. So, and also, let's say, myosin, right? Technically, it has 2 functions. It binds to ATP and it binds to actin, even though both are for muscle contraction. Should it be considered multifaceted or not?
Okay, so good question. I think that the major problem that we also discussed together yesterday is that everybody has its own specific definition of what is a protein function. And the problem become more and more complex talking about multifunctionality. So our idea was to collect all the different kinds of multifunctionality all together to create the widest data set. And in particular, about the enzymes problem, the fact that an enzyme has multiple substrate is the strict definition of what is promiscuity. So we decided to include this kind of enzymes, and we try to collect them and retrieve them directly. So yes, they are present, and we consider them multifunctional. I don't know if it's answered your question, and I hope that maybe the data set will be enlarged in the future with many different proteins, as long as the multifunctionality has been assessed in the literature. So yes, try to find your protein. It is not present in the actual release, it will be in the next one, and please email me if something is missing, because it was a manual curation. As you know, the human protein has so many, so maybe we will increase the number of the data set.
Great talk. Really have to hold these close. Okay. My question is about the tissue distribution. It's more about the hypothesis that might underlie this. So one thing that when I looked at this, you said there's no signal, and that might be true statistically, but I'm like, all of these multifaceted proteins look like they're more expressed than their companions. So one hypothesis is that the multifaceted proteins might play different roles and might be expressed in like more tissues, for instance. Did you look at that? Like whether or not they were multifaceted proteins were expressed in more tissues than their counterparts?
Yes, we believe that this could be the case, but we didn't actually go further in this analysis. There is a little signal, yes, almost everywhere. This could be also a bias, maybe because multifaceted proteins coid be more studied in deep than the other proteins. So we need to be more strict and have more analysis on that. But yes, this is a good idea.
Thank you, Giulia. Wonderful.
No, thank you.
And I love seeing it all over the years. So, the question, I mean, I have many questions, but one question is, do you see that these multifaceted proteins are older? So is it that they acquire additional functionality? And then the other sort of related question is, are these functions related by simply changing the ligand, let's say, or some reaction partner? And it's really not that much of a change, except maybe a broadening.
Okay, so I will start answering the second question because it's easier. In this case, yes, as I told you in the previous answer, there are some proteins that just change maybe the substrate, or maybe change just the ligand. And there are also families of proteins that are not so specific in terms of the ligand. So yes, maybe it would be the case. And we decided to collect them as multifunctional, but we checked that in the literature they are addressed as multifunctional with multiple different tasks. And there are also proteins, like in the case of moonlighting proteins, in which the same domain have completely different function, like in the case of aconitase or other different proteins, then depending on the conditions, will change completely in their function, from enzymes to binding DNA, RNA, whatever. So completely different function. About a signal in the ancient proteins compared with newly developed function, this is an issue. We would like to study indeed maybe some protein families for which we have many data, also to study when the second function has been acquired during the evolution, but we didn't do it yet.